Hello and welcome back to Investigator Insights, targeting the c-Met receptor as a strategy for cancer therapy. Joining me once again are Dr. Johanna Bendel and Dr. David Spiegel from the Sarah Cannon Research Institute here in Nashville, Tennessee. So David, in the last session we were talking about the importance of protein overexpression of the c-Met receptor and the value of immunohistochemical staining in identifying patients whose tumors were MET high as opposed to MET low. And you mentioned that in one of the trials with onartuzumab that patients in whom their staining was high, 2+ or 3+, definitely seem to benefit as far as progression-free survival and overall survival. But if they were MET low, they actually did worse when onartuzumab was added to standard therapy. So how important in the future, going forward, do you think the role of immunohistochemical staining is going to be? What is going to be the role of FISH as far as gene amplification? And are there other potential biomarkers, such as Johanna had mentioned, HGF overexpression, that you see potentially being important in the personalization, an overworked term, but the personalization of therapy in non-small cell lung cancer?
Yeah, I mean, I think it's complicated right now. We're waiting on results from trials. As you point out, there are many ways to measure alterations in MET, and even at our center, we're doing next-generation sequencing now, where we can identify point mutations in MET. It would be nice to think that IHC is going to be the way to do it, because that seems to be the cheapest and most readily accessible way to do it. But we don't really have any standards in place for what that will be. I mean, we have trials where it was done a certain way, but I've told you an example of onartuzumab, the way it was done in that study. But that's not the way it's being done with rituximab, an important drug that's being developed in gastric cancer. So what if both trials are positive, and yet both use different IHC methodologies to measure MET? You know, how is that going to play out with companion diagnostics and requirements by the FDA? I think we don't know. It'd be nice to think that IHC is going to be a major way of assessing MET status. I think that's one to bet on. But I think FISH is important, and we know certainly from the onartuzumab data that is important, that certainly the patients who have higher degrees of amplification match up with patients who have the highest expression, and they seem to do the best. Numbers are small, but suggest that trend that the more you have of this target, the better you do with a drug that inhibits that target. But I think it's fair to say we don't know. And one quick word about the ligand story, HGF. There's been some difficulty in figuring out how to measure that. The actual antibody test that helps you measure that can attach to free ligand and cause the number to go up. So just the assessment of it can make the levels of HGF seem higher. So that has not been entirely worked out.
Interesting. When we talk about the clinical trials, and some trials are using progression-free survival, it's a primary endpoint. Some are using overall survival, safety, and other factors, including overall response rate, are looked at. With these targeted agents, where there are often more cytostatic than cytotoxic and tumor destructive, what do you think is the best endpoint to really measure this? Is it measuring a biomarker, or is progression-free survival a better endpoint? Is overall survival a realistic endpoint? So, Johanna, I'm gonna pick on you with
this. Thanks for giving that to her.
Yeah. So I mean, we talked about in the Marquis trial, overall survival endpoint not having been met. But with patients getting so many different lines of therapy, you really wonder, is overall survival a realistic endpoint? It's probably the easiest to measure, but is it always a realistic endpoint?
You know, I think it depends on the tumor that you're looking at. I mean, for something like HCC, overall survival is probably very realistic. Even gastric cancer, probably very realistic. Colon's becoming messier, breast, lung becoming messier. But what's very interesting in looking at the randomized phase 2 studies that have been presented so far with MET inhibitors, what I find very interesting is that the overall survival data seems to be much more pronounced in terms of benefit than the progression-free survival data for these studies. And so I'm not sure where we might not be causing some sort of intrinsic change within the tumor. Because typically when you see a benefit in progression-free survival, that same degree of benefit, if you have a benefit in overall survival, holds. But in the MET story, it seems to increase. And so why that is, I'm not sure. I don't know that anybody's sure. So does that make the cell eventually more responsive to whatever subsequent therapies it gets? Is this more play on the cytostatic potential? Are we measuring progression in the wrong way? This is a big matter of debate right now within a lot of the targeted agents, is what is disease progression, and are we measuring it correctly with just basic RECIST? So I think right now within the trials that are ongoing, we have to capture progression-free and overall survival to see if this trend continues. Response rate, even though we used it with the ralatumumab data in combination with panitumumab, doesn't seem to be as much of a player within the MET story, so I don't know that I would rely on those results as much as I would some sort of survival result.
Very good. Any comments, David?
Yeah, I think that's fair. It gets hard to lump everything together. You know, breast and certainly FDA, requirements, so to speak, have been different, right, than in breast than they have been for lung. In colon, colon probably has the strongest data that PFS is a good surrogate for OS, but I think it's different. It is concerning, right, when you have a drug like crizotinib or erlotinib that have these dramatic response rates. In fact, that's what led to crizotinib's approval. You see a drug work like that, you think it's, that's got to be a real drug, that's really hitting the target. And you're not really seeing that so far with the therapies we've discussed today. Is that a reason for concern? And I agree with Johanna that it may not be that's the best measure of these drugs' activity, and maybe OS is the only way to show that. But it is a little frustrating that you're not seeing those waterfall plots like you see with those other targeted drugs.
So what do we know about resistance? I mean, most of these agents, when they have a positive effect, it lasts for 6 months, sometimes 12 months, and some situations even longer. But eventually, the tumors develop resistance. What do we know about resistance mechanisms against the c-MET targeted agents?
To be frank, I think we know very little. I think we're still working on trying to figure out if it's a realistic target. I guess we know that, but is it going to be a clinically do we have drugs that can be effective clinically that can to suppress this target? And I think, like with erlotinib and crizotinib, just to follow up on that, or trastuzumab, I think then we start to learn more about why patients become resistant. There is work going on now, and we know, you got into this in the beginning in our first segment, that there's all this interconnectedness with other pathways. So we know that other families went signaling. We know EGFR. We know angiogenesis may play a role too, and why this pathway may not stand alone, and that there may be other escape mechanisms. Certainly, we know PI3 kinase is well known in this regard. And it may be, and you're getting at this, I suspect, that an anti-MET therapy may not be the best or only strategy here. We may have to think about combining that with other targeted therapies.
Yeah, I think it may turn out this is more of a co-driver than the primary driver. It's the co-pilot. Well, let's go on and talk about the ongoing phase 3 trials, and we'll move back to GI malignancies. Tell us about some of the phase 3 trials with these novel agents and where things stand, and maybe we can help drive accrual to those trials.
Right now, there's two major phase 3 trials that are ongoing. The first one is in hepatocellular carcinoma, looking at tivantinib plus or minus placebo in second-line therapy. And this is taking off of that randomized phase 2 that we discussed in the earlier segment, where we saw that benefit in time to progression and overall survival, giving a MET inhibitor blocker. So that's one that's occurring currently. The other one is the onartuzumab gastric study that's open. And this is looking at FOLFOX plus or minus onartuzumab for first-line metastatic gastroesophageal cancer treatment. So those are the two large randomized phase 3s that are.
So these are HER2 non-overexpressors.
So these are only HER2 non-overexpressing tumors. So you do need to know that status before enrolling.
Talk about a disease crying for a new therapy.
Yeah. Yeah, so we're very excited. Most of the other studies right now are randomized phase 2s, and we're at the point now within GI malignancies where we're taking those randomized phase 2s and we need to look at the data. So we've had some press releases. For instance, there was the cetuximab irinotecan plus or minus tivantinib that was press released as not meeting its primary endpoint of progression-free survival. However, we still, as with the tivantinib non-small cell lung cancer study, we still haven't seen the data yet. Did MET status play an effect? Did previous treatment play an effect? So we have to see some of those results before we can carry those forward. We're also waiting on the results from the accomplished FOLFOX bevacizumab plus or minus onartuzumab study. So as we start to see some of these results come in, I think we'll start to get more of an idea of what's going on. There is one accruing randomized phase 2 study that I wanted to talk about very briefly for hepatocellular patients. And this is a study that is looking at sorafenib plus or minus E7050, which is a combined c-MET VEGFR tyrosine kinase inhibitor that's out there. And I know that hepatocellular trials are very difficult to accrue to. And so this is one that definitely makes a lot of sense for hepatocellular patients. So that is opening and accruing right now.
Very good. So that'll be almost like combining sorafenib with the c-MET target. Very, very interesting. Well, David, what about in non-small cell lung cancer? We talked earlier about the Marquis trial. It failed to meet its primary endpoint. At least it appears that it would. But there's still probably a lot of interesting data subset analysis that hopefully will come out of that trial. What other trials are going on?
So right now the only major, I guess, phase 3 trial is the onartuzumab trial. It's the follow-up to the phase 2 study. This is we touched on it briefly in the second segment, but this is a trial that is aimed at improving overall survival in patients with refractory non-small cell lung cancer.
The MET lung.
The MET lung in the second and third line setting. So these are patients who are randomized to onartuzumab or erlotinib alone. To get on this trial, and this is the difference from the phase 2, you have to have a MET high-expressing tumor. So the way this works is patients are screened, and actually we were allowed to screen patients in the first-line setting. So you could have a patient on carbo-Alimta, for example, who you screen for MET status. You send that into a central laboratory, and the turnaround tells you that your patient is, yes, eligible for the study or not. You actually get EGFR mutation results with that as well. And then those patients who are high scored the same way as in the phase 2, 2 to 3 plus and greater than 50% of the cells. Those patients are then eligible for randomization in the second and third line setting. So there are many patients actually in screening, or we call pre-screening, who we already know are high and are waiting. In fact, just recently, we closed pre-screening because the commitment to those patients needs to be honored if they're waiting to go on to those studies, to that study. For other patients who we find out they've progressed today and they're looking at this as an option, they can still enroll in this trial. It's just that you have that little bit of wait where you have to send the tissue in, see if you're going to be a high expressor or not. It's early. We're waiting to see. Is this really a 50% cutoff? Do you have a 1 in 2 chance of getting onto this trial, as the phase 2 trial suggests? And I think it'll be interesting to see what the actual prevalence is of high expression. But that trial is ongoing. We expect that trial to finish enrollment by the end of the year here, and then look to our outcomes thereafter. Hopefully, in the next 2 years, we'll have results.
Very good. What about some of the other novel agents that also include c-Met in their target array? You mentioned crizotinib, cabozantinib. I think there's another antibody called ficlatuzumab that is out there. How are those agents coming along?
Well, I'll just say quickly, in lung cancer, it's slow but steady development. I think ralutumab is a very exciting drug, and I'm wanting to see more of that in lung cancer. There's some small studies that are being looked at in lung cancer, but I think the gastric data is what everyone's waiting on.
To interrupt really quick, because that was the third randomized phase 3 I forgot to mention in my last. So there is also a gastric ralutumab phase 3 going on.
But then to get to your excellent point about crizotinib, right? A drug that was first developed, Johanna knows this very well, first developed as a MET inhibitor, later quickly transitioned into an ALK inhibitor drug, but now Pfizer is going back and looking at its role in MET inhibition. So those are early trials, still waiting on results. And then in the prostate world, as you mentioned, the recently approved drug from Exelixis, if I'm going to say it,
cabozantinib.
Cabozantinib, is that its trade name? I can't recall. So that drug's interesting, right?
I never remember the trade names.
That's interesting, right? That's a drug that is, I think many refer to as a dirty drug, right? Because it inhibits a lot of things, like sunitinib may be responsible for more than just anti-VEGF properties, right? MET is one of the targets of that drug, and the data so far have been early in lung cancer. So there's been a small series of patients who have evidence of a response to a drug like that. But that drug is not yet into any large-scale lung cancer trials that I'm aware of. I'm sure those are being planned right now.
No, and I think right now I agree with David. I'm hoping that crizotinib will start moving into the GI arena as well. A cabozantinib, not quite there. I think that there was some attempted development of a precursor compound to cabozantinib, for lack of a better term, called XL880, which was a combined VEGFR c-Met receptor tyrosine kinase inhibitor, where they looked in gastric cancer quite a few years ago and didn't see any signal really come through, and so that's why I'm not sure that they've gone down that pathway into GI malignancies, but hopefully this is just the start of something new. There's a lot of other malignancies that are being targeted right now as well by MET inhibitors of various sorts, and triple-negative breast cancer is one of the tumor types, thyroid cancer, renal cell carcinoma. So I think the MET inhibitors are here to stay in all their various forms.
Right, just a lot of interesting data hopefully will come pouring out at national meetings in the next year or two. Well, very good. This has been really interesting. I have enjoyed it. I've learned a lot, and I hope our viewers have also. And thank both of you for taking the time to come and provide us all this really amazing information.
All right, thanks.
Thank you, and thanks to all of you for watching this segment of Investigator Insights, targeting the c-Met receptor as a strategy for cancer therapy. So please check all of these 3 parts. I think you will learn a lot about this very interesting emerging target for cancer treatment. Thanks again.